Okay. So good morning. It's great to see you again. So today what I'm going to talk about is phase-appropriate GMPs for investigational new drugs. So I'm going to start with providing you with some background, defining what GMP means, and the regulatory framework around GMPs, including statutory requirements, regulations, and guidances. Then I will try to put together the relationship between CMC, GMP, and quality systems, and finally describe what are outline what are the GMP expectations for all the phases of development, starting with research and development, preclinical, phase 1, 2, 3, and then briefly describe inspections. So what are GMPs? GMP stands for Current Good Manufacturing Practices, and these are regulations that are enforced by the FDA. They provide persistence that assure the proper design, monitoring, and control of the manufacturing processes and facilities. And adherence to GMP assure that the product has the identity, the strength, the quality, and the purity by basically controlling the manufacturing process and the operations. So it is expected that you have robust quality systems, that the raw materials are monitored, the quality, that you record the quality of the raw materials that are introduced into the manufacturing process, that there are that you detect deviations to your manufacturing process and you investigate them, and that is also recorded. And then that testing in the laboratories makes sense, that the assays that you use are suitable, that the results are recorded too. So the idea is that the quality is built into the design of the manufacturing process and is not just assured through end testing of the product. So we talked about current good manufacturing process, and the current word there means basically that the GMPs are flexible so that manufacturers can decide how to implement the best controls for their processes. And also invites to innovation, to using up-to-date technologies and innovative approaches so that you keep your systems up to date. So something that you may have implemented 20 years ago that was up to date at that moment might not be adequate now. For example, I don't know, to control viruses or contamination or cross-contamination. So there is a regulatory framework around the GMPs. GMPs are based on the law, on the statute, and then FDA has issued regulations to provide more details on how to apply the act. And then there are some guidances that describe the current thinking on GMPs. So what are the legal bases for GMPs? Section 501 of the Food, Drug, and Cosmetic Act requires the drug are compliant with GMPs, and basically a drug will be deemed or shall be deemed adulterated if the methods, the facilities, the controls, the manufacture, the processing, the packing, all the operations around that drug are not do not conform with current good manufacturing practices. This is, so FIDASHA, that is an amendment to the 501 Act that was implemented in 2012, expanded the GMP concept by adding the oversight and control over the manufacturing process. So these are the laws that establish the GMPs, and then there are regulations, and the regulations are basically there to explain how to implement the statutory requirements. The regulations for GMPs are set forth in the Part 210 of the CFR 21 and Part 211 for biological products. In addition to the 210 and the 211, the 600 to 680 apply as pertinent, and GMPs also apply for investigational drugs. That is what we are talking about now. So for phase 1 material, phase 1 manufacturing, or the manufacturing of a phase 1 drug should comply, must comply with the statutory GMP requirements. However, they are exempted to comply with the 211 CFRs, and that is put forward in section 210C, that states that an investigational drug for use in a phase 1 study is exempted from compliance with the regulations in Part 211. But not all the phase 1 drugs are exempted from the 211s. These are the two exceptions. When the phase 1 drug has been used already for another indication, for example, in a phase 2 and a phase 3 study, then they should be compliant with the phase 3 expectations for GMP. And the same if you use a marketed drug for a phase 1 study, the expectations for that drug are really full GMPs because it's a marketed drug. A more detail regarding the expectation for CGMPs in a phase 1 are put forward in a guidance, FDA guidance, that is current good manufacturing practices for phase 1. It's outside if you want to have a look at it. And basically, the idea of this, so the 211s are mostly, they apply mostly to commercial products. And so if we would use that rigor for phase 1, we might never initiate a phase 1. So the idea is that there are GMPs in place that ensure the safety of the human subjects, but also facilitate the initiation of a phase 1. And then for phase 2 and phase 3, full 210 and 211 are expected. So yesterday I talked to you about the fact that the CMC information is graded through development. So there should be sufficient information in each stage of development, and this sufficient information depends on what is the product, what is the study, what are the number of people enrolled. So CMC is graded, so it's phase dependent. GMPs are also phase dependent, and then there is another key player that are quality systems. The quality systems support the development and manufacture of investigational drugs, and adherence to these quality systems assure the safety and quality of products intended for clinical use. And this is done by appropriate monitoring and control of the whole process that is used to manufacture the product, so that, again, we come to the consistency in the manufacture, so that you have a manufacturing process that is going to deliver a product of consistent quality, so that you can build all the information that you gather through phase 1, 2, and 3 until you get to commercial. The implementation of the quality systems is also graded, and they are mostly evaluated during an inspection. So phase appropriate GMPs that are compliant with the quality system ensure that you generate CMC information that is appropriate to meet our expectations for the different stages of development, and also follow the recommendations set forth in the guidances. So the CMC information is going to be submitted in the dossier, and we are going to review it. And basically, there is going to be also recorded in GMP documents in your facility. The GMP part of safety of a drug is mostly evaluated by FDA inside, in the inspection. But ultimately, these two very important parts of product quality come together through the quality systems. And there are some areas that overlap. So, for example, process validation, you are going to submit all the data for process validation in the dossier. We are going to review it, but then we are also going to look at it when we go on inspection. So, again, GMPs are phase dependent, and although during research and the development phase, the GMPs do not apply in their strict sense, they should be so mostly during this stage you are collecting data, you are getting knowledge, and so you need to have a way to manage that knowledge so that it doesn't that is not lost. And so the data and the information should be documented and traceable, and that includes all the data and information that relates to the production of the development lots, of the toxicology lots, the characterization and testing of these toxicology lots, as well as records of the raw materials that were used during these stages. And this is really important specifically for the raw materials that are animal-derived or are of biological origin, so that you, when you look back, you know where the risks were, and you work on them and put forth controls to mitigate them. This, I think, it was, for example, important when the BSc started, so that everybody could kind of trace down where in their process they had used some raw material that could be at risk of bearing BSc. And the important thing is that all that information is going to ultimately provide support to the clinical material. So it's very important that that information is recorded. And then it's going to be very useful to ensure that the process is scalable and transferable to a GMP facility without impact to the CQAs. So adherence to GMPs for phase 1 generally include the existence of well-defined and written procedures, adequate control of the equipment and the manufacturing environment, and accurately and consistently record of data that comes from the manufacturing and testing. So when you manufacture a phase 1 material, it's very important that you consider the hazards that are and the risk associated from manufacturing the product in a certain environment. For example, you may use a facility that is shared by other products, and you may need to make sure that there is no cross-contamination of your product, that your product is not going to be contaminated with other chemicals that were used during manufacturing or during research activities in that facility. So it's very important that a thorough evaluation of the facility where you're going to manufacture your phase 1 material is done, and the risks are identified and mitigated. So basically, the idea is that once you identify, for example, you share an equipment with other projects, and so make sure that the material, the equipment is clean, and so that your phase 1 material is not at risk. So it's important to take appropriate actions prior to and during the manufacturing of the phase 1 material to eliminate or mitigate potential hazards and risks. And these actions must be in effect for the manufacturer of each lot of the investigational drug used in the phase 1 clinical study. And I think this came a little bit in the previous talk. Both the sponsor and the manufacturer are responsible for assuring that the phase 1 material is manufactured under GMPs. So what are the manufacturing controls that apply to a phase 1 investigational drug? So they refer to personnel. The personnel has to be trained, sufficiently trained to perform the activities that they have to. There should be a quality control functions, and these functions should be clearly written, and the responsibilities should be separate from manufacturing. The person or the group of persons that determine the release of a lot should not be the same that manufacture the lot. So the ones that trigger investigations or implement CAPAs should not be the same. The same person should not be doing the same both activities, ideally. The facilities and equipment, the facilities should be adequate to manufacture a product that is going to go into humans. And so there should be adequate working areas, adequate equipment. The equipment has to be calibrated to provide relevant results. Cleaning is important. The light, the air conditioning, the filters, where you are going to prepare your material has a lot of impact on its safety. There should be control of the components in the container closure, so there should be written procedures that determine what are the requirements for the container closure that you are going to use, what are the acceptance criteria, that make it suitable for its intended purpose. Manufacturing records, there should be clearly a clear description of how the manufacturing was done. If there was a deviation to the planned manufacturing process, then that should be clearly clearly shown in the records. If, for example, when you add the worksheets for the testing results, there should be assurance that there is some kind of traceability of those records, that it's not that you can print out a paper and write something down. So this kind of control is very important because it's going to ultimately help you build up on very, very solid grounds. The same with laboratory controls. It's important that the methods that are used in the labs are suitable, the results that are reliable, that they are controlled, that the results are recorded, and that samples are retained, because especially stages, but especially in early stages, you may need to retest. And so it's very important that if possible, you have retained samples. And then it's important to start, as I think I mentioned yesterday, that it's very important to generate instability data very early. The same with packing, labeling, and distribution. It's very important that the packaging where you transport your material to the clinical side is adequate, that you have written procedures on how to handle it, how to pack the drug so that it's protected. And again, record keeping is key. Document control. I mean, you will hear me saying about document and record a lot, because really for phase 1 and for early stages, that is one of the most important aspects of GMP. So data and information should be recorded. This includes release data, in-process testing, raw material qualification, manufacturing data, and also all the changes to the procedures or processes and a rationale for those changes. So again, being able to track back, how did you do things, what did you use to manufacture, so that when you come back, all the information is there. This, the information should be fully traceable to primary or source data. There should be systems in place that ensure that the information is not vulnerable to alteration, and the retention of complete and accurate data is a GMP requirement. So for multi-purpose facilities, there are additional considerations that should be taken that ensure that there is no cross-contamination between your product and other products that might be also manufactured in that facility. Of course, manufacturing should happen one product at a time. There should be appropriate cleaning procedures and control procedures in place so that to ensure no carryover of materials or products or mix-ups. And then this also impacts the facility. The facility should have a design that ensures or mitigates cross-contamination. For biological products, it's very important to take into account the potential for adventitious agents. From the product perspective, that the product is not contaminated with adventitious agents from the environment, and also some products might be risky for, for example, spore-forming. You may be producing your phase 1 material or your material in spore-forming microorganisms. So it's important that could be then released to the environment. That has to be controlled too. And a very key aspect of GMP is for parenteral products to ensure sterility. This product is a phase 1, but it's going to go into human subjects. And so the accepted manipulations should be performed in aseptic workstations. There should be media fields, process simulation using bacterial growth media, so that we ensure that you are going to feel that your material is going to be sterile. Environmental monitoring for the same purpose, and then the material should only be released after sterility testing is completed and it's negative, or it's positive for sterility. And with regard for GMPs for phase 2 and phase 3, well, it's a risk-based approach of applicability of GMPs during development. Some GMP aspects start very early in development, and then they continue throughout the development. That is, for example, testing for bioburden, testing for endotoxin, training. You always want the operators to be trained to do the task that they have to do. Calibration of instruments, that is really expected from the very beginning. There are some other aspects that vary with the phase. For example, process validation. You would not expect process validation in the phase 2, in phase 3. You generally have do it like during phase 3 and towards licensure. And then, well, yeah, that is more for the phase 2 and phase 3. So method validation is something that you are going to kind of increment through the phase development. And compliance with 21 CFRs Part 211 are expected for phase 2 and phase 3. And basically, this kind of wrap up, hopefully, all the concepts that we have discussed so far. And so as knowledge on the process and understanding of the process and the product evolves, then the GMPs become more stringent. And this is what we call the graded aspect of the phase-dependent GMPs and CMC. So they both go together. Early in development, you're going to have experience with one lot. Maybe you have an instrument. Your testing is limited to a lot. I mean, very few lots. So really, the GMPs are going to be the same, but less complex. But as you manufacture more, then you may need more training for employees, more equipment. You may go into different facilities. So all this comes together, and together with the increment in the knowledge in the CMC, the GMPs then also become more stringent, until you get to the point that you have a manufacturing process that is feasible for a commercial setting. Inspections. There is no requirement for inspections of IND clinical material manufacturers. The facility, however, we can inspect a facility at any time. There could be for cons inspections, and FDA might decide to conduct an inspection. But we seldomly do that. And because of that, adherence to GMP is generally the adherence to GMP during development is generally evaluated in a pre-license inspection. So this is a case study in which there was a submission of a BLA for a new biologics, and then we scheduled a pre-license inspection for the drug product facility because that drug product facility had never been inspected by FDA. So because of that, the sponsor or the manufacturer hired an external consultant to evaluate the readiness for inspection. And this consultant identified gaps in the documentation control system. So they found that the systems were vulnerable to manipulation, and they also found evidence of data manipulation and deficiency in data quality oversight. So the question then became how accurate the data that we had in the BLA, how accurate the data were. So during the review cycle, all the data needed to be reevaluated, look for traceability, accuracy, and impact on those lots that ultimately were going to establish product safety and efficacy. And then the sponsor had to implement adequate CAPAs to identify the vulnerabilities of the document control systems and mitigate the risk in future manufacturing. Well, I think I went through all this. You might read it. This is a list of resources, and I am done. So if you have any questions, please let me know.
Online? Offline? Sorry. Well, yeah, online. Sorry.
In what phase would you definitely want to have your CGMPs ready?
Well, I guess I try to emphasize the fact that they are graded. So for sure for commercial, that's for sure. But when you manufacture the phase 3, I think that you have to have strong GMPs are going to be the data that support commercial too.
My phone number, nice and close.
So my question is about, does FDA allow to use, the vectors contains ampicillin-resistant gene for recombinant protein expression?
Can you start from the beginning? I didn't understand.
Okay. So my question is about, does FDA allow, to use vector contains ampicillin-resistant gene for recombinant protein expression and production for phase 1?
If we allow that you have a gene, what kind of gene?
Ampicillin.
Ampicillin. Oh,
yeah.
ampicillin. Well, I don't think so. We strongly recommend that no antibiotics are used for fermentation at least steps. So maybe in the early, but those are maybe for tetracycline. I don't think that.
What if, I mean, so, okay, so what if, you know, the ampicillin does not involve in the fermentation stage?
No, I understand what you're saying.
Yeah, but before the fermentation, say, you know, I'm preparing the expression plasmid DNA. So, you know,
Oh, for the DNA, so for cloning.
Yeah, for cloning.
Just for cloning.
Yeah, for those.
So you're not going to use it for anything else?
No.
I think that could be appropriate, but I can come back to you because I know that ampicillin is not antibiotic that we would like, but it's kind of in the cloning vector. I don't think that is an issue.
Okay, thank you.
Microphone number 1.
I've got a question about personalized medicine, and in particular, how it might relate to a multi-purpose facility that's manufacturing more than one drug at a time. If you're making a personalized product in the context of phase 1 and have adequate controls in place, can more than one lot that's patient-specific be manufactured at the same time, or does there need to be complete clearance before?
So these are different products for different patients? Yeah.
So each lot would be for...
My assumption is that you may need to do kind of a
Thank you.
Yeah.
All right, let's take another online question if we could. Just.
When does the FDA require the analytical methods to be validated during the drug development?
So validation of analytical method is expected for licensure.
Okay, microphone number 2, please.
Yes. FDA prefer to using GMP material for the IND enabling studies and phase 1, or you still prefer that the IND enabling study just using a so-called engineering law of GMP, not a GMP law. Or my question to you is, I would prefer, if we can afford it, better using all GMP products for toxicology.
Let me see if I understand you. So your question is whether you can use a non-GMP for the toxicology, and then for phase 1, obviously it has to be GMP.
Yeah.
So the question is whether you could use, we would prefer a toxicology lot to be manufactured under GMP?
No, my question in other way around.
Okay.
So would you prefer, to avoid the comparable and consistency
And that you use
issue?
the very same lot?
Yeah, just using the same law for GMP.
That is your
law.
decision. That is really your decision. Whether, I mean, it's better for you to manufacture one GMP lot and use it in the toxicology studies and the clinical studies. I think
It's better.
That is what is better for you, because sometimes it's easier for the sponsor, the manufacturing, to really do something non-GMP and test it in the tox studies before moving into a GMP. But that is ultimately something that you might
Great.
want
Thank
to
you.
try. My pleasure.
Okay, and if you have questions in the room, come on up to the microphone, but let's go with our next online question, please.
Are manufacturers of atypical actives required to follow CGMPs as described in the ICH?
Manufacturers, no?
Can you repeat it one more time?
Are manufacturers of atypical actives required to follow CGMPs as described in the ICH?
Atypical what? I didn't understand atypical.
Atypical actives, active ingredients. It didn't say active ingredients, but it says
yes.
actives.
I think I come back to this, to what I try to convey, that is basically a phase graded GMPs. ICH generally are for licensed product when you get to licensure.
And for those of you online, thank you for keep your questions coming in, but you don't get bonus points for speed. So take the time to read it back to yourself to make sure it sounds good before you hit submit. Let's take our next online question for me, if you would.
Is a drug intended for 505(b)(2) submission considered phase 1? In other words, if a sponsor is doing a bioequivalent study with its availability bioequivalent study with its test drug versus versus an approved RLD, does the test drug need to follow full CGMPs?
I am not familiar with bioequivalence studies. If someone can chime in, I do mostly BLAs. My guess is yes, but
We don't seem to be getting any takers in the back of the room.
yeah.
That'll be a great question to resubmit to CDER
Yeah, please.
SBIA at fda.hhs.gov. We've got just a couple minutes left. If there's any questions in the room, come on up to the mic. Let's take one more question from online.
When there's a site change for a phase 1 IND, what information must be submitted to the IND for that particular site change?
So it depends on the product, but I guess so. I guess that the question implies that the phase 1 was performed or is ongoing. So basically, the information again is data, and we are not going to evaluate GMP in a phase 1 IND through the paper. So basically, it's raw data, tabular data, all the data that provides us some assurance that there is adequate documentation of the information, and of course that the data supports comparability. That is like evaluation of the data per se.
Okay, and it looks like we can squeeze in one more question from online, please.
Can you give an example for the additional requirements of CGMPs that are there when drug development moves from phase 1 to phase 2 and 3?
Well, as I mentioned, for a phase 1, you would not expect compliance with the 211 and 212. When you move into a phase 2, then the GMPs should really increase the stringency, and the expectation is that they are kind of compliant. They are compliant with 210 and 211. That is not expected for phase 1.
All right. And with that, that's all the time we have for questions. Please help me thank Cecilia for coming to talk with us.